Hi students, welcome to a new semester and our first learning video. I wanted to just provide a little bit of background on why we're starting the class with the subject of the scientific method. I have a couple of major course-long learning outcomes for you. The first is to increase your curiosity, which can then lead to more creative thinking and the ability to reason or problem solve. These are important life skills, and the scientific method is a great framework for learning how to do this. Most people teach the scientific method in a linear form, and we'll discuss some alternatives to this. But to begin our thinking on the scientific method, the first step is observation. In other words, noticing an interesting phenomena. Again, it's allowing that curiosity to drive us to notice the things that are happening around us, particularly in the natural world. We begin to see patterns that activate our thinking. We start to form questions. And when we have a tentative answer to a question in mind, we form what's called a hypothesis. Once we have a hypothesis, we then develop an experiment to test that hypothesis. And after we've tested the hypotheses, we perform data analysis and hypothesis testing. What's interesting is that those tests can fail, and often they give us information on what we don't know. And when we know what we don't know, we can refine our observations, rehypothesize, retest, and reanalyze. So the scientific method is far from linear, it is iterative. Just a little side note, we as humans have an incredible capacity to take limited information and draw conclusions or connections from it. This is called heuristics, and it's incredibly powerful and valuable when we're in danger and we take limited information to make quick decisions. But it can create misguided thinking, erroneous thinking, and bias when we apply heuristics or mental shortcuts to understanding science. So, science and the scientific method is designed to avoid bias. And we do that in science by using what we call hypothesis testing. I'm going to give you a quick example of hypothesis testing. So, in the scientific method, first we make observations and we have questions. We form hypotheses, which is the tentative answer to a question. And then we create what is called an alternative hypothesis. For example, we're walking along, we observe that some flowers appear to be taller where it's sunny. That would be our alternative hypothesis. We often just shorten this to the word hypothesis, but it's our best guess at explaining the observation that we've made. Now, to avoid any potential bias because of this ability that our brain has to take limited information and draw connections and conclusions, we always in science want to counterbalance alternative hypotheses with what we call a null hypothesis, which is the opposite of the alternative hypothesis. The alternative hypothesis and the null hypothesis are mutually exclusive, meaning both of them cannot be correct. And that is by design. So in this case, a null hypothesis in this situation would be that the sun does not affect the flower growth. Then we would initiate an experiment and collect and analyze data to see which of these two hypotheses is supported by our data. So let's jump into a case study so we can think about this in the context of a real world application. This is an observation that my son Isaac made when he was very young as we were doing research down in the Mojave Desert. Our son noticed something. He said, "Why are all these dead insects on the tips of these very sharp banana yucca leaves?" And as he was observing this pattern, he could see that a lot of the impaled insects were flying insects, and that in some cases their wings were kind of splayed open as they lay dead on these sharp points of leaves. So, from his observation, a question began to form in his mind. How and why were these insects getting impaled on sharp leaf tips? And as he thought more about it, an alternative hypothesis formed in his mind because of the wings and kind of splayed patterns of the wings. Because of his experience in the Mojave Desert, where it's oftentimes very windy, his alternative hypothesis was that he thought the wind was blowing these insects onto these sharp points, and that's how they were dying. As we talked about it, he checked himself by forming a null hypothesis, which was that the insect impaling themselves had no effect on or was not affected by wind at all.
So there are two types of experiments that we could design to test these two competing hypotheses. The first is a natural experiment in which we can leverage naturally occurring events to study the effects of one factor on another. So, for example, we could go and look at a number of different sites across the Mojave Desert and measure the wind speed. If there are large differences in wind speed, then we measure the number of dead insects at each site. We could look at the relationship between wind speed and insect impaling. In natural experiments or natural studies, the researcher or the scientist doesn't do anything to control the conditions in the environment. You're simply looking at the natural variation in the environment and what you measure. The second type of experiment is simply called an experimental study. In this design, the researcher controls the experimental conditions. So, for example, in this case study, we could take this screen cloth right here and create low wind speed conditions next to an area where there isn't this cloth as a wind control. And then we would compare the two areas. This is an experimental design where the researcher controls the conditions of the experiment.
To analyze this data, we're looking at how the explanatory variable, that's what we're manipulating, in this case, the windscreen or no windscreen, influences the response variable, what we're measuring, or the number of impaled insects. Once we've conducted the experiment, then we take the data and we begin to plot it or graph it out. So let me just orient you on the graph because we're going to be looking at lots of graphs to interpret hypotheses that we generate over the course of the semester. There are generally two axes on graphs. There is the x-axis or the x label and the y-axis or the y label. And what we put on the x-axis is always what we manipulate in the study. In this case, it would be wind speed. In a natural experiment, it would be just measuring the different wind speeds at different locations. And in the controlled experimental study, it's the wind cloth versus the non-wind cloth as an experimental control. So the x-axis always explains what we're measuring, or what's changed on the y-axis. There are two different types of plots that we'll look at in this class. So I want to orient you to what they look like. The first is what we call a scatter plot. And the second is what we call a bar plot. The main difference between the two, you'll notice, is what we put on the x-axis. So for the scatter plot, we put continuous data on the x-axis. So in this case, this natural experiment, we've gone to dozens of sites, we've measured wind speed, and then we've charted out the wind speed across the x-axis. And then at each site, we've also measured the number of dead insects. And you'll notice that our x-axis data is continuous data, meaning it's numbers. And over here in the bar plot analysis, our x-axis is categorical data. It's data in categories or groups using word labels. Now I'll explain how we generate the statistics to interpret the data and to determine which of our hypotheses is supported in this case study, our alternative hypothesis or our null hypothesis. So again, here is our natural experiment on the form of a scatter plot. And in both analyses, we have the same thing that's been measured, impaled bugs. What is fundamentally different is what we find on the x-axis. So in this case, what we see is that we have dozens of data points. We charted out the wind speed at each site and the number of dead bugs that we found. And that creates a pattern in the data. That's what we call the scatter plot because the data scatters on the x and the y axis. And then statistically, the next thing that happens is you take a linear function. So this line right here, and you run it through the scatter plot with the goal of minimizing the distance from the data point to that line. And then to calculate what's called the R squared value, you measure the distance from each point to that line. So the more that the data are scattered, the farther they are away from the line, the lower is the R squared value. If all the data points are on the line perfectly, then you'll get the highest R squared value possible, and that is 1, which means that 100% of the variation in what you're measuring is explained by what's on the x-axis or the explanatory variable, in this case, the wind speed. So our R squared value is very, very low. The data are scattered far from the line, and it doesn't appear like there's any strong relationship between wind speed and impaled bugs. And the way that we interpret this 0.02 R squared means that 2% of the scatter or the variation in our data can be explained by wind speed. And this p-value that's associated with this data is 0.88, meaning it's very, very unlikely that there's a significant relationship between these two variables. There's an 88% chance that there's not a strong statistical relationship of wind speed on impaled bugs. So this is a strong support in our natural experiment for our null hypothesis that wind is not related to or influencing bugs being impaled on these sharp leaf points. And over here in our experimental study, we have our cloth that we put up to block the wind, and then nearby, in the absence of the cloth, we just have the wind blowing through. And in this case, again, we measured impaled bugs in areas where we have wind cloth and not. And then we've measured this maybe dozens of times and in dozens of sites, and we take all of this data and we average them to get a mean value. In this case, under the wind treatment at the 40 different sites, we saw an average of about 10 impaled bugs at each site. And at the no wind treatment, where we had the wind cloth, it was 10 impaled insects as well. So the mean difference are very, very small here. The other thing we have to measure, though, is how consistent the data are across all those sites. So we see that by these error bars, and they're very large, which means the data are inconsistent. So some sites might have 2, some sites might have 12 or 14, and this gives us less confidence that we can distinguish between significant effects between the two treatment conditions. And then using the differences in the mean averages and the amount of variance, the variability of the data, the calculated average across the sites, we generate the probability or p-value. The p-value tells us how likely our statistics are to give us a clear picture of what's going on in our experiment. And in this case, it gave us a p-value of 0.94, which tells us we're about 94% confident that there's no difference between these two variables. Or in other words, there's only a 6% chance that there is a meaningful, significant difference here between the treatments. So we have very, very low confidence that there's any difference between wind and no wind in terms of impaled bugs. So we conclude that our experimental study supports the null hypothesis that wind is not impaling the bugs.